Polymerase chain reaction. The polymerase chain reaction, in short which is called the PCR, is a biomedical technology in molecular biology used to amplify a single copy or a few copies of a piece of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. Developed in the 1983 by Kary Mullis, PCR is now a common and often indispensable technique used in medical and biological research labs for a variety of applications. These include DNA cloning for sequencing, DNA-based phylogeny or functional analysis of genes, the diagnosis of hereditary diseases, the identification of genetic fingerprints used in forensic sciences and paternity testing, and the detection and diagnosis of infectious diseases. In 1993, Mullis was awarded the Nobel Prize in Chemistry along with Michael Smith for his work on the polymerase chain reaction. The method relies on thermal cycling, consisting of cycles of repeated heating and cooling the reaction for DNA melting and enzymatic replication of the DNA. Primers, which are short DNA fragments containing sequences complementary to the target region along with the DNA polymerase after which the method is named, are key components to enable selective and repeated amplification. As PCR progresses, the DNA generated is itself used as a template for replication, setting in motion a chain reaction in which the DNA template is exponentially amplified. PCR can be extensively modified to perform a wide array of genetic manipulations. Almost all PCR amplifications employ a heat-stable DNA polymerase such as the TAQ polymerase, an enzyme originally isolated from the bacterium. This DNA polymerase enzymatically assembles a new DNA strand from DNA building blocks, the nucleotides, by using single-stranded DNA as a template and DNA oligonucleotides, also called DNA primers, which are required for initiation of DNA synthesis. The vast majority of the PCR methods use thermal cycling, that is, alternately heating and cooling the PCR sample through a defined series of temperature steps. In the first step, the two strands of the DNA double helix are physically separated at a high temperature in a process called DNA melting. In the second step, the temperature is lowered and the two DNA strands become templates for DNA polymerase to selectively amplify the target DNA. The selectivity of PCR results from the use of primers that are complementary to the DNA region targeted for amplification under specific thermal cycling conditions. PCR is used to amplify a specific region of a DNA strand, the DNA target. Most PCR methods typically amplify DNA fragments of between 0.1 and 10 kilobase pairs. Although some techniques also allow for amplification of fragments up to 40 kbp in size. The amount of amplified product is determined by the available substrates in the reaction, which become limiting as the reaction progresses. A basic PCR setup requires several components and reagents. These components include DNA template that contain the DNA region target to be amplified, two primers that are complementary to the 3' ends of each of the sense, an anti-sense strand of the DNA target, TAQ polymerase or another DNA polymerase with a temperature optimum at around 70 degrees centigrade, deoxynucleoside triphosphates, sometimes called deoxynucleotide triphosphates, nucleotides containing triphosphate groups, the building blocks from which the DNA polymerase synthesizes a new DNA strand, buffer solution providing a suitable chemical environment for optimum activity and stability of the DNA polymerase. Bivalent cations, magnesium or manganese ions. Generally, magnesium ions is used, but manganese ions can be utilized for PCR mediated DNA mutagenesis, as higher manganese concentration increases the error rate during DNA synthesis. Monovalent cation potassium ions. The PCR is commonly carried out in a reaction volume of 10 to 200 microliters in small reaction tubes 0.2 to 0.5 milliliter volumes in a thermal cycler. The thermal cycler heats and cools the reaction tubes to achieve the temperature required at each step of the reaction. Many modern thermal cyclers make use of the Taylor effect, which permits both heating and cooling of the block holding the PCR tubes simply by reversing the electric current. Thin-walled reaction tubes permit favorable thermal conductivity or allow for rapid thermal equilibration. Most thermal cyclers have heated lids to prevent condensation at the top of the reaction tube. The procedure. Typically, polymerase chain reaction consists of a series of 20 to 40 repeated temperature changes called cycles, with each cycle commonly consisting of 2 to 3 discrete temperature steps, usually 3. The cycling is often preceded by a single temperature step at a high temperature which is above 90 degrees centigrade and followed by one hold at the end of the final product extension or brief storage. The temperatures used and the length of the time theory are applied in each cycle depending on a variety of parameters. These include the enzyme used for DNA synthesis, the concentration of divalent ions and the DNTPs in the reaction and the melting temperature of the primers. The initiation step only required for DNA polymerases that require heat activation by hot start. This step consists of heating the reaction to a temperature of 94 to 96 degrees Celsius or 98 degrees Celsius if extremely thermostable polymerase are used, which is held for 1 to 9 minutes. The denaturation step. This step is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees centigrade for 20 to 30 seconds. It causes DNA melting of the DNA template by disrupting the hydrogen bonds between complementary bases, yielding single-stranded DNA molecules. The annealing step. The reaction temperature is lowered to 50 to 65 degrees centigrade for 20 to 40 seconds, allowing annealing of the primers to the single-stranded DNA template. This temperature needs to be low enough to allow for hybridization of the primer to the strand, but high enough in order for the hybridization to be specific, that is, the primer should only bind to the perfectly complementary part of the template. If the temperature is too low, the primer could bind imperfectly. If it is too high, the primer might not bind. Stable DNA-DNA hydrogen bonds are only formed when the primer sequence very closely matches the template sequence. The polymerase binds to the primer template hybrid and begins DNA formation. The extension or the elongation step. The temperature at this step depends on the DNA polymerase used. TAQ polymerase has its optimum activity temperature at 75 to 80 degrees centigrade, and commonly a temperature of 72 degrees centigrade is used with this enzyme. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding DNTPs that are complementary to the template in the 5th primer to the 3rd primer direction, condensing to the 5th phosphate group of the DNTPs with the 3rd hydroxyl group at the end of the nascent or the extending DNA strand. The extension time depends both on the DNA polymerase used and on the length of the DNA fragment to be amplified. As a rule of thumb, at its optimum temperature, the DNA polymerase will polymerize 1000 bases per minute. Under optimum conditions, that is, if there are no limitations due to limiting substrates or reagents, at each extension step the amount of DNA target is doubled, leading to exponential geometric amplification of the specific DNA fragment. Final elongation step. This step is occasionally performed at a temperature of 70 to 74 degrees centigrade. This is the temperature needed for optimal activity for most polymerases used in the PCR. For 5 to 15 minutes after the last PCR cycle to ensure that any remaining single-stranded RNA is fully extended. The final hold. This step at 4 to 15 degrees centigrade for an indefinite time may be employed for short-term storage of the reaction. Let us now look at the polymerase chain reaction stages. The PCR process can be divided into 3 stages. The exponential amplification. At every cycle, the amount of product is doubled, assuming 100% reaction efficiency. The reaction is very sensitive. Only minute quantities of DNA needed to be present. The leveling off stage. The reaction slows as the DNA polymerase loses activity and as consumption of reagents such as the dNTPs and primers cause them to become limiting. The plateau stage. No more product accumulates due to exhaustion of reagents and enzyme. Let us now see PCR optimization. In practice, PCR can fail for various reasons, in part due to its sensitivity to contamination causing amplification of spurious DNA products. Because of this, a number of techniques and procedures have been developed for optimizing PCR conditions. Contamination with extraneous DNA is addressed with lab protocols and procedures that separate pre-PCR mixtures from potential DNA contaminants. This usually involves spatial separation of PCR, setup areas from areas for analysis or purification of PCR products, use of disposable plasticware, and thoroughly cleaning the work surface between reaction setups. Primer design techniques are important in improving PCR product yield and in avoiding the formation of spurious products, and the usage of alternate buffer, components or polymerase enzymes can help with amplification of long or otherwise problematic regions of DNA. Let us now study about the application of the polymerase chain reaction. Selective DNA isolation. PCR allows isolation of DNA fragments from genomic DNA by selective amplification of a specific region of DNA. This use of PCR augments many methods, such as generating hybridization probes for southern or northern hybridization and DNA cloning, which require larger amounts of DNA representing a specific DNA region. PCR supplies these techniques with high amounts of pure DNA, enabling analysis of DNA samples even from very small amounts of starting material. Other applications of PCR include DNA sequencing to determine unknown PCR amplified sequences, in which one of the amplification primers may be used in Sanger sequencing isolation of a DNA sequence to expedite recombinant DNA technologies involving the insertion of a DNA sequence into a plasmid or the genetic material of another organism. Bacterial colonies, E. coli, can be rapidly screened by PCR for correct DNA vector constructs. PCR may also be used for genetic fingerprinting, which is a forensic technique used to identify a person or organism by comparing experimental DNAs through different PCR based methods. Amplification and quantification of DNA. Because PCR amplifies the regions of DNA that it targets, PCR can be used to analyze extremely small amounts of sample. This is often critical for forensic analysis when only a trace amount of DNA is available as evidence. Quantitative PCR methods allow the estimation of the amount of a given sequence present in a sample, a technique often applied to quantitatively determine levels of gene expression. Quantitative PCR is an established tool for DNA quantification that measures the accumulation of DNA product after each round of PCR amplification. PCR in diagnosis of diseases. Polymerase chain reaction permits early diagnosis of malignant diseases such as leukemia and lymphomas, which is currently the highest developed in cancer research and is already being used routinely. PCR allows for rapid and highly specific diagnosis of infectious diseases, including those caused by bacteria or viruses. PCR also permits identification of non-cultivable or slow-growing microorganisms such as mycobacteria, anaerobic bacteria or viruses from tissue culture assays and animal models. The basis for PCR diagnostic applications in microbiology is the detection of infectious agents and the discrimination of non-pathogenic from pathogenic strains by virtue of specific genes. Viral DNA can likewise be detected by PCR. The primers used need to be specific to the targeted sequences in the DNA of a virus, and the PCR can be used for diagnostic analysis or DNA sequencing of the viral genome. Variations on the basic PCR technique. Assembly PCR or polymerase cycling assembly. Artificial synthesis of long DNA sequences by performing PCR on a pool of long oligonucleotides with short overlapping segments. The oligonucleotides alternate between sense and anti-sense directions, and the overlapping segments determine the order of the PCR fragments, thereby selectively producing the final long DNA product. The next is the digital PCR. The digital PCR used to measure the quantity of a target DNA sequence in a DNA sample. The DNA sample is highly diluted so that after running many PCRs in parallel, some of them will not receive a single molecule of the target DNA. The target DNA concentration is calculated using the proportion of negative outcomes, hence the name digital PCR. Helicase dependent amplification. Similar to traditional PCR, but uses a constant temperature rather than cycling through denaturation and annealing extension cycles, DNA helicase, an enzyme that unwinds DNA, is used in place of thermal denaturation. The next is hot start PCR. This is a technique that reduces non-specific amplification during the initial setup stages of the PCR. It may be performed manually by heating the reaction components to the denaturation temperature, example 95 degrees centigrade, before adding the polymerase. Specialized enzymes have been developed that inhibit the polymerase activity at ambient temperature, either by the binding of an antibody or by the presence of covalently bound inhibitors that dissociate only after a high temperature activation step. Hot start cold finish PCR is achieved with new hybrid polymerases that are inactive at ambient temperature and are instantly active at elongation temperature. In silico PCR, digital PCR, virtual PCR and electronic PCR refers to computational tools used to calculate theoretical polymerase chain reaction results using a given set of primers to amplify DNA sequences from a sequenced genome or transcriptome. Silico PCR was proposed as an educational tool for molecular biology. Inverse PCR. This is commonly used to identify the flanking sequences around the genomic inserts. It involves a series of DNA digestions and self-ligation resulting in known sequences at either end of the unknown sequence. Multiplex ligation dependent probe amplification. This permits multiple targets to be amplified with only a single primer pair, thus avoiding the resolution limitations of multiplex PCR. Multiplex PCR. This consists of a multiple primer sets with a single PCR mixture to produce amplicons of varying sizes that are specific to different DNA sequences by targeting multiple genes at once. Additional information may be gained from a single test run that otherwise would require several times the reagents and more time to perform. Annealing temperatures for each of the primer sets must be optimized to work correctly with a single reaction and amplicon sizes. That is, their base pair length should be different enough to form distinct bands when visualized by gel electrophoresis. The next is the nested PCR. This increases the specificity of DNA amplification by reducing background due to non-specific amplification of DNA. Two sets of primers are used in two successive PCRs. In the first reaction, one pair of primer is used to generate DNA products which besides the intended targets may still consist of non-specifically amplified DNA fragments. The products are then used in second PCR with a set of primers whose binding sites are completely or partially different from the located primers used in the first reaction. Nested PCR is often more successful in specifically amplifying long DNA fragments than the conventional PCR, but it requires more detailed knowledge of the target sequences. The next is overlap extension PCR or splicing by overlap extension, a genetic engineering technique that is used to splice together two or more DNA fragments that contain complementary sequences. It is used to join DNA pieces containing genes, regulatory sequences or mutations. The technique enables creation of specific and long DNA constructs. It can also introduce deletions, insertions or point mutations into a DNA sequence. The next is quantitative PCR. This is used to measure the quantity of a target sequence, commonly in real time. It quantitatively measures starting amounts of DNA, cDNA or RNA. Quantitative PCR is commonly used to determine whether a DNA sequence is present in a sample and the number of its copies in the sample. Quantitative PCR has a very high degree of precision. Quantitative PCR methods use fluorescent dyes such as the EVA green or the fluorophore containing DNA probes such as TOCMAN to measure the amount of amplified product in real time. It is also sometimes abbreviated to RT-PCR, which means to say real-time polymerase chain reaction, but this abbreviation should be used only for reverse transcription PCR. The next is reverse transcription PCR. For amplifying DNA from RNA, reverse transcriptase reverse transcribes RNA into the cDNA, which is then amplified by the PCR. The RT-PCR is widely used in expression profiling to determine the expression of a gene or to identify the sequence of RNA transcript including transcription start and termination sites. If the genetic DNA sequence of a gene is known, the RT-PCR can be used to map the location of exons and introns in the gene. The next is thermal asymmetric interlaced PCR. For isolation of an unknown sequence flanking an unknown sequence. Within the known sequence, tail PCR uses a nested pair of primers with differing annealing temperatures. A generate primer is used to amplify in the other direction from the unknown sequence. The next method is the touchdown PCR. A variant of PCR that aims to reduce non-specific background by gradually lowering the annealing temperature as PCR cycling progresses. The annealing temperature at the initial cycles is usually a few degrees, 3 to 5 degrees centigrade above the Tm of the primers used. While the later cycles, it is few degrees, 3 to 5 degrees below the primer Tm. The higher temperatures give greater specificity for primer binding, and the lower temperatures permit more efficient amplification from the specific products formed during the initial cycles. With this, we come to the end of polymerase chain reaction. Thank you.